Right, good morning everyone. Welcome back. So obviously in the last few weeks we've been looking at the innate immune system. So what we're going to do now is we're going to move on to have a look at the adaptive immune response. And today we're going to sort of focus on an introduction to the B cell receptor and the T cell receptor and have a look around those sorts of things. So the learning outcomes for today, that by the end of the lecture we'll be able to describe the key differences between the innate and the adaptive immune system. So this is something that we've been building on over the last few weeks and we'll continue to build on this. And you'll also know about the T cell receptor, so the TCR and the BCR, their structure, and also the relevance of co-receptors in propagating that signal across the membrane. And then also understand how we get that diversity within the T cell receptor and the B cell receptor. So first of all, let's just do the attendance pin, which is 1629. So that's 1629. Okay, so this is obviously a picture of hematopoiesis, and this is something that we've seen before. And it's just really to illustrate the fact and remind you that the cells of the innate immune system are derived from the common myeloid progenitor, whereas when it comes to the adaptive immune system, these are generated from the common lymphoid progenitor. So we have our T cells, our B cells, and then these can further differentiate. And hopefully you remember that from the first week. And this was actually a little task that I kind of set you to kind of work on as we go through the lectures. So this was comparing and contrasting the innate and the adaptive immune response. And I've put in here a few hints in case you've been struggling with that, but hopefully you can build on that. But the one thing that I wanted to pick out, essentially today, is that when it comes to the innate immune response, you remember that the receptors are actually germline encoded, whereas with the adaptive immune response, we have a further set of systems, and these include somatic hypermutation, and these are things that we will build on in the next set of lectures. And also, just to refresh your memory, that the innate and the adaptive immune response are interlinked and they collaborate with each other. So when we get an infection, we have the innate immune system kicking in, and if this isn't cleared by the innate immune system, we'll then get the adaptive immune response taking over and hopefully clearing that infection. So the innate immune system kicks in first. If it's not cleared by that, we have the activation of the adaptive immune response. Remember that the innate immune system relies on these PAMPs. So we've got our pattern recognition receptors recognizing these patterns. Now, when it comes to the adaptive immune response, we have a much broader pathogen-specific recognition, and this is mediated via our antigen receptors, so our B cell receptor and our T cell receptor. So the adaptive immune response isn't using pattern recognition, it's using something that's much more specific and can be tailored. And when we get this first exposure to antigen, what we get is this clonal expansion. So we expand the specific B cell or T cell that can recognize that unique antigen so that we can then mount an infection against that. And also at the same time, we propagate this long-lived memory that allows us to maintain that sort of memory of that pathogen if we ever should encounter it again. So when it comes to the adaptive immune response, a lot of these interactions are actually occurring within the lymph node. So this is just a schematic of the lymph node. So lymph nodes will lie at the junctions between lymphatic vessels, and they're kind of broken up into several key areas. So these blue areas here are the T cell areas where the T cell resides, and then the B cells will reside in these yellow areas. And these are actually germinal centers. So when you get an infection, you'll start to get this formation of a germinal center, and obviously if you have an infection, the germinal center will be quite large, and so you can see that here on the staining of this cross-section. And this is where, in the germinal centers, that this fine-tuning of our antibody response will actually take place. But essentially, the lymph node is kind of a loose organization of leukocytes. And this is all connected by a lymphatic system. So this is obviously just a little bit of a refresher because we've already come across this before. But cells of the immune system can, they'll leave the blood via these small capillaries and then they'll enter the lymph tissue. If they don't encounter a pathogen, they will leave the lymph and they will have, essentially will have this lymphocyte recirculation going on, but if they do encounter a pathogen, then they will stop recirculating and we'll get this antigen presentation going on. So just really to stress the fact that the lymph node and the secondary lymphoid tissue, they're very dynamic, they're always in a state of flux, in a state of change, so it's not a static environment. And then also just to stress the fact that the innate and the adaptive immune response are interlinked, and just to show this diagram again, because we want to know how does the antigen actually get into the lymph node. So remember, it's via these antigen-presenting cells, and in particular the dendritic cell, which is acting as a cross between the innate and the adaptive immune response, and is presenting that antigen to your B cells or your T cells. So it's the dendritic cells that are one of the professional antigen-presenting cells. So we continue to look at dendritic cells. So they were first discovered by Ralph Steinman, and he initially wanted to call them claudiocytes, so this was actually after his wife, I believe, but that obviously didn't happen, and they were called dendritic cells. So remember that the dendritic cell, it's recognizing those pathogen-associated molecular patterns via its PRRs, and we then get processing of this antigen and this presentation to T cells, and this is actually something that we'll cover on in the future. And that will help to sort of instruct and tailor the adaptive immune response. But the experiment that this man actually did to show the importance of the dendritic cells, he took a pathogen and he also took lymphocytes from a spleen and he mixed them together. And what happened? Nothing. Okay? So what he then did is he did the same thing. So we've got our lymphocytes with the pathogen, but this time we've got the dendritic cell in there. And this time we mounted an adaptive immune response. So what it's important to take home from this is that T cells don't actually recognize entire pathogens. What they're recognizing is the small bits of pathogens that have been chopped up and presented to them by the dendritic cell. And if we look at that experiment in a little bit more detail, so this is just the same experiment that we discussed, but if we now take lymphocytes and we mix them with a dendritic cell, and we also take lymphocytes and a dendritic cell, and this time we mix them with peptides that are chopped up, what might happen, do you think? So in this response, if we've got lymphocytes and a dendritic cell, we have no response. But this time, if we're adding in bits of a peptide, so it's almost like a vaccination in a way, what do you think will happen? Will we get a response? Actually, we don't. Because the dendritic cell needs to see the entire pathogen, okay? It needs that trigger from the PAMP, so the pathogen or the pathogen-associated molecular patterns. And in a way, that's actually known as the immunologist's dirty little secret, that even with a vaccination, that's not enough. You need to actually trigger the dendritic cell via its PRRs. Okay? It needs to see that pathogen or that pathogen-associated molecular pattern. So that's why when you have a vaccination, the vaccination is often mixed with what's called an adjuvant. And so that will actually trigger the innate immune system as well. So in this scenario, you've stepped on a dirty nail. You've got some bacteria invading your body. This is detected by the dendritic cells, which will then migrate to the lymph nodes, and they will then present this antigen to T cells. Okay? So this is the link between the innate and the adaptive immune response. So if we look at antigen, because it's the antigen receptors of the B and the T cells that will actually recognize this antigen and gives us that specificity. So here we have antigen in the red, and you can see that antigen, it can have different epitopes on it, which is sort of summarized in this diagram here, or we can have the same epitopes. Now, when we say an epitope, this is really a group of chemical structures or amino acids that are actually exposed on the surface of a protein or a molecule, and it's the epitopes that will actually generate that antigenic response. Okay, so we can have different epitopes or similar epitopes. Now, when it comes to an adaptive immune response, Now, we're looking here at B cells and T cells, but for the sake of it, let's just say that this is a T cell, because the T cell are really the sort of key players, the master controllers of the adaptive immune response, if you like. So, obviously, we have a whole range of T cell clones, well, basically different specificities. They all have a unique antigen receptor on them. So when we encounter a pathogen, that pathogen will probably be recognized by one of those T cells out of the whole mixture of different T cells that we've got. But obviously, that's great. We've got a T cell that can recognize that pathogen, but that's just one cell. Okay, so that's not really going to help us. So what we need to do is we need to clonally expand that one cell so that we have a whole army of cells that can recognize and fight that pathogen. So that expansion is called clonal expansion. So when a particular T or B cell encounters the pathogen via its antigen receptor, it will become activated, and then it will proliferate, and we get that clonal expansion, so that each clone is able to recognize and eliminate that specific pathogen that it recognized initially. So they're all a clone of each other. And we'll come on to that in a few slides' time as well. So as I mentioned, antigen is recognized by the antigen receptor, and we have antigen receptor on our B and our T cells. So on the T cell, we have a specific receptor that's called the T cell receptor, or the TCR, whereas on the B cell, we have the B cell receptor, and that's abbreviated to BCR. And that's just showing them in these two diagrams here. So we have the B cell receptor, and we have the T cell receptor. So B cell receptor, you can see it's this antibody shape. So essentially, it is membrane-bound immunoglobulin, okay? And that B cell, when it comes to secrete antibody, it will secrete antibody that's exactly the same, identical to its membrane-bound receptor. We have a slightly different structure for the T cell receptor that we'll look at. But it's an important note to take home, is that if you try and look at a B cell and a T cell under the microscope, they're going to look exactly the same. You're not going to be able to determine that. The difference is, obviously, that they've got the B cell or the T cell receptor on their surface. So if you do things like FACS staining or fluorescent activated cell sorting, you can identify them that way. But just looking at them under the microscope, under staining, you're not going to see a difference. So just comparing them in a little bit more detail. So obviously we have a B cell here. It's got its membrane-bound immunoglobulin. If it becomes activated and it differentiates into a plasma cell, which is obviously the job of a plasma cell is to secrete antibody, we will get secretion of antibody, again, exactly the same as the membrane-bound BCR. Also, you'll notice that the B cell receptor, it has two antigen binding sites, so it's got one on this arm of the antibody and one on the other arm. Now, if you contrast that to the T cell receptor, this has a slightly more simplistic structure, but it just has one antigen binding site. And we have, essentially, the antigen binding site is made up of these variable regions. So it has differences in the amino acid structure, and this creates a really sort of vast array of potential binding sites that allows it to recognize a vast array of potential antigens and therefore pathogens. So let's sort of focus now on the B cell receptor. So as I've already mentioned, antibodies are the secreted form of the B cell receptor for that specific cell. Now generally, surface-bound B cell receptor is known as immunoglobulin, but that term is a little bit more fluid. So when you see immunoglobulin, it also refers to antibody now. So you're not going to be, let's say, corrected if you call antibody immunoglobulin, but traditionally immunoglobulin was surface-bound B cell receptor. So the B cell receptor will bind epitopes on extracellular antigens. That's important to remember. They don't work intracellularly. So they're particularly good, let's say if you have a viral infection and that virus has been released outside of the cell, that these antibodies can bind that virus, and they can block it and prevent it from entering neighboring cells and propagating that infection. And they can also help to clear virus by binding to it in immune complexes, and then that can be eliminated from the body. So there's a little bit of a difference that's quite important to stress between B cells and T cells. And this is that the B cell will recognize antigen in its native form. so whether it's carbohydrate, protein, or lipid, whereas the T cell will only recognize peptides that have been processed and then presented on an antigen-presenting cell. Okay? So the B cell will recognize native peptides or proteins. So obviously the function of the B cell is to produce antibody. So you can see here we've got our B cell receptor with its membrane-bound, the B cell with its membrane-bound immunoglobulin. So this is the B cell receptor all over the surface. If it encounters a pathogen or a bacteria to which it can recognize, so it can recognize the epitopes on the bacteria, what happens is that B cell will become activated. As we already talked about, it will proliferate and we'll get that clonal expansion and we'll get differentiation into plasma cells. And these plasma cells will then secrete antibody that's of the exact same specificity as the B cell receptor. And that antibody will then obviously bind to that bacterium. And then, as we talked about in the last few lectures, this can then trigger the complement pathway because the antibody will act as an opsonin. And then that complement pathway can eliminate that pathogen. And also we can get promotion of phagocytosis. So just kind of linking that in to what we talked about last week. So in terms of B cell receptor antigen binding sites, remember that it's recognizing that whole protein in its native form. And the part of the antigen that the antibody will recognize is referred to as the epitope. And we can see here that antigens, they can be multivalent, so you can see here it's all got the same epitope, or an antigen, as we mentioned, can have different epitopes. Now, when it comes to that antibody binding to an antigen, we can see that the epitope, it can be linear, or it can be discontinuous. So in discontinuous, it's binding to that peptide or that protein, in its native form. So you can see the three-dimensional structure. So obviously in terms of research, if you were using an antibody to recognize a protein and you're doing it via Western blot or something like that, you obviously have to boil and heat inactivate, denature your proteins. So, not all antibodies are going to work in the same way. So just remember that this is obviously binding it in its three-dimensional structure. So they can recognize the whole proteins in a variety of different manners. If we start to look at the structure of the antibody, so obviously it's this Y-shaped molecule, and we had a little look at this in the last lecture. So we have here our heavy chain. So you can see in this instance, the heavy chain is running here. We've got two heavy chains, and then we've got two light chains, which are shorter. Okay. We have a variable region. So this is the antigen binding site here. So we have a variable region, which gives the antibody that unique specificity to bind to its antigen. And then we have constant regions, which are obviously constant, so they're not variable. And these constant regions are shown in blue. Now, at the antigen binding site, we have this complementary determining region. So this is what binds to that antigen and gives it its specificity. And then at the C-terminus, we have the FC region. So remember that if we were to cut the antibody up, we would have two Fab fragments. So these are fragment of antigen binding, and then the fragment crystallizable, which is the C-terminus down here. And it's the C-terminus of the antibody that will interact with various effector molecules. So remember that in red are the variable regions, which give the antibody that unique structure. The blue regions are the constant regions. So the antibody is binding to the antibody binding site on these variable regions, and we have two heavy chains, so these are the longer chains here, and then we have a light chain that's stuck onto the side. Okay? And we've got two antigen binding sites, one on each arm, and they're identical in terms of their ability to bind to antigen. Now, if we look at the B cell receptor, so this is membrane-bound immunoglobulin, we have some co-receptors as well. And the important co-receptors for the B cell receptor are this Ig alpha and Ig beta. So these are co-receptors, and what they do is they help to mediate that signaling of the B cell receptor. So you'll see that they have these very long cytoplasmic tails, and this helps in sort of propagating that signaling across the membrane. Okay, so the antibody will bind up here, and obviously you don't need to go into too much detail, but you just need to be aware that these are the co-receptors that help to sort of transmit that signal from the B cell receptor across the cell membrane, and they sort of facilitate that intracellular signaling. Now, if you look at any given B cell receptor or T cell receptor, essentially they're going to look the same when you just glance at them, but obviously each one will have a very unique specificity when it comes to what antigen it will detect. Now, once a lymphocyte has been generated, it may well never come into contact with its cognate antigen, because we have a huge array of lymphocytes with different B cell receptors or T cell receptors. We will have ones that can recognize antigen that may not even naturally exist, and we may well never come into contact with it. But if a lymphocyte does come into contact with its cognate antigen, as I mentioned, it will become activated, it will proliferate, and you'll get that clonal expansion to produce a whole army of identical clones. And this is known as the clonal selection theory, and this was actually proposed in the 1960s and the 1970s. And so if we look at that in a diagram, so here we have a whole host of different, let's say, B cells in this case. They've all got different B cell receptors on their surface with unique specificity to a given antigen. If we have an infection with a pathogen, we can see that this lymphocyte here can actually recognize antigen on this pathogen. So that's great. We get that proliferate, we get that activation, that proliferation, that expansion, so the clonal expansion. Because remember that one cell on its own is not going to be good enough to do any kind of job, so therefore we need to expand it, so we've got this army of identical clones. Now, some of them will differentiate into plasma cells, which will secrete that antibody, which can then go and target that pathogen and promote its phagocytosis, etc. But a proportion of them will remain as memory cells, because that's great if you've secreted antibody and you've fought off the infection, but you want to maintain a memory of it. Because obviously if we ever encounter that pathogen again, then we need to be able to respond quickly and a little bit more quickly. So some of the B cells will remain as memory B cells should we ever encounter that pathogen again. But this led to a problem that was called generation of diversity problem, also known as the GOD, or the God problem. So in the 1960s and the 1970s, the immunologists and the researchers were very convinced by this clonal selection theory. But there was a little bit of a problem as to how we can generate so many different antibodies. So in humans, we have around 25,000 genes. So this, would give us a limited number of receptor specificities if you think about it. We've got a set number of genes. How can we make so many unique antigen receptors? Because in our bodies, we will have antibodies, against chemicals that don't even naturally exist. So that's kind of a bit of a unique situation. And essentially, this was solved in the 1980s when we have this Japanese researcher, so this is Susumu Tonegawa, and he got the Nobel Prize for discovering VDJ recombination. So this is the genetic mechanism, which is a series of recombination events that allows us to produce this antibody diversity. Okay? So in VDJ recombination, the lymphocytes use gene segments, so they recombine gene segments to enhance the genetic variability. So from a limited number of genes, we can get a very large number of receptors. So we can have a large outcome from a very small select number of genes. And the VDJ just stands for variable, diversity, and joining. And these are the name of the segments that get put together in VDJ recombination. So VDJ recombination is what essentially gives us that huge diversity to be able to mount an immune response against an antigen that we've never even seen yet. So the number of antibodies that can be produced is essentially limitless. And what happens is the receptor genes from the lymphocyte are organized differently to other genes, and we'll see that. So it's somatic as opposed to germline encoded, if you were to compare that to the innate immune system. So we have these whole families of gene segments that can be recombined, and these gene segments are what is inherited through the germline. But these segments need to be rearranged first, and this will take place in the developing B cells during the bone marrow. When it comes to T cells, they actually leave the bone marrow and then they finish their development in the thymus. But essentially, the VDJ recombination process is the same when it comes to B cell receptors and T cell receptors. So they undergo essentially the same kind of process. So if we look at that in a little bit more detail, so you can see that for B cell receptors, is what we're looking at here, there's several different steps. We have this combinatorial diversity, so this is where we're getting those segments recombined. We're getting junctional diversity, so this is where we're getting nucleotides being added and subtracted at the junctions where we're putting these segments together. We're then getting combinatorial diversity of the antigen binding site, so we're putting together different combinations of heavy and light chains. So remember on the diagram of the antibody that we've got a heavy chain and a light chain. And then we also have, so this is all occurring in the bone marrow, but then in the lymph node we have this somatic hypermutation. So just introducing this now, but we'll talk about it in more detail at a later lecture. And this only occurs in B cells once they've been activated by antigen. So we'll just start to run through these first three steps here and just show you what's going on. So this is VDJ recombination. So we're looking here at the locus for the light chain and the heavy chain. So the light chain locus is highlighted in blue. You'll actually see that we've got two light chain locuses. We've got the lambda and the kappa. And this is located on chromosome 22 and chromosome 2. And then for the heavy chain locus, this is located on chromosome 14. But you'll see for the light chain locus, we have V segments, and we have J segments, and we have a constant region as well. When it comes to the heavy chain segment, you'll see that we have the V segment in red, we have the D in green, and the J in yellow, and then a constant region as well. Okay? And this is obviously just summarizing that structure. So in green, we've got the heavy chain, and then in blue, we've got the light chain. And so it's just showing you, obviously, that the light chain, this is encoded for by either the lambda or the kappa, and the heavy chain is encoded via chromosome 14. And this is where we have the VDJ. So VJ for the light chain and VDJ for the heavy chain locus. And this table is really just showing that this sort of random recombination of the gene segments via VDJ recombination really produces this massive diversity that we have for our antigen receptors. So we start to look now at the actual mechanism of VDJ recombination and how that works. So this is showing the process for the light chain and the heavy chain. So here we have our germline DNA at the top. If we look at the light chain, what happens is we start to get recombination between the V segment and the J segment. So we now get this VJ segment. This is then combined with constant region so that we can form our light chain. So here we have our light chain with our variable region and our constant region. When it comes to the heavy chain, again up here we have our germline DNA. Initially here we get DJ recombinations, so these are coming together. And then the DJ is coming together with the V, so we're getting VDJ recombination here. These are then brought together with the constant region such that we can actually generate our heavy chain. And then obviously the heavy chain and the light chain will be combined to form that immunoglobulin. So these segments are all being cut and respliced together. Yeah?
Question about the light chain. Why is there no D segment in the variable region?
That's just the way it is. Don't really have an answer for that one, but yeah, that's just how it works. So these segments are all being cut and spliced together, and this is being mediated by these recombination signal sequences, or these RSS. Now, this is showing an RSS here. So actually, the RSS is composed of 3 elements. We have a conserved heptamer, so 7 base pairs, and a conserved nonomer, so 9 base pairs. And in between that is either a 23 or a 12 base pair spacer sequence. Okay, so you can see one RSS here with a 23 spacer and another one here with a 12. And the way that they're arranged dictates that a 12 base pair RSS must pair with a 23 base pair RSS. So that's known as the 12-23 rule. Okay, and we'll look at that in a little bit more detail on the next slide. But these are being cut and spliced together by essentially the VDJ recombinase. And the VDJ recombinase has a couple of important activating genes, and these are known as RAG1 and RAG2. And these are the specific enzymes that will play a part in that VDJ recombination and actually sort of splicing the DNA, so cutting the DNA and splicing it together. So you can see here we've got our RSSs. So what will happen is RAG will come along and it will bind to the RSS sequences that it's going to combine. So you can see here that RAG is binding to this 12 base pair RSS, and it will also bind to this 23 RSS that's further down. So what we want to do in this instance is we want to join this V segment with this J segment. So we want to remove what's in the middle. So the RAG will bind here, and it will also bind to the other RSS. And what this does is it mediates this hairpin structure, so this looping out of the DNA that we don't want. So you can see that we wanted V1 and J brought together. So it's brought V1 and J together, and this intermediate DNA is looped out. Other components of the VDJ recombinase will cleave the DNA, and then we'll get a recombined coding joint that will form. So that ultimately what we'll end up with is the coding joint of the V1 and the J segments brought together. So you can see over here in the original diagram, they were quite spaced out, but the RAG has bound to them, brought them together. The hairpin structure that's been looped out has been cleaved. The coding joint is joined back together to form our successfully recombined VJ segment. And then we have a signal joint, which is the intermediate bit that we don't want, which will be left over and then degraded. So this is the essential process by how all these segments are being put together. And obviously this is just an example for V and J, but this will be going on all the way throughout the different steps so that you can essentially bring together the different segments for your unique receptor. So that was the combinatorial diversity, so bringing together the different V, D, and J segments. But then we have this junctional diversity and this nucleotide, this non-template nucleotide addition. So what we're doing here is we're bringing in random sort of nucleotides at these junctions, at these gene segment junctions. So this is quite a little bit of a complicated diagram, but it's quite easy to go through. So obviously here we've got our RSSs, so our heptamers in this instance. RAG will have bound to the RSSs and it will have cleaved the DNA, so you can see that at these arrows here. So what will happen is RAG will initially nick one strand of the DNA, and then this kind of loops out, as you can see here, and this creates this palindromic sequence. So you can see how it's been nicked here on one side, it's opened out, we've got a palindromic sequence on one strand and another one on the other strand. And these are known as the P nucleotides, so P for palindromic, okay? So you can see we've added in some extra base pairs. Then what happens is we have the addition of N nucleotides. And this is mediated by an enzyme called TDT. And so this enzyme will come in, and it will add on these extra nucleotides. So we've got our P nucleotides, and we've now added on some N nucleotides to that as well. And then we'll get pairing that's occurring. So we'll look to match these up. Once we get pairing, which you can see in this diagram here, we'll get removal of any unpaired nucleotides. So that what we'll have now is this small junction, but we'll have a bit of a gap in terms of the DNA because obviously it's not been paired completely, but we'll get DNA synthesis that will actually fill in any gaps, and so we'll get this repaired junction here. So what you can see is that from the end of the junction of these two gene segments that we started with at the beginning, we've got this whole addition of this random sequence now in between the two segments. And that will give sort of extra variability and an extra uniqueness to that antigen receptor, okay? That's a good question. I'm not entirely sure. I think probably because you've got base pairing, I think it's probably there's some flexibility in that, but I'm not 100% sure, okay? So by this process, by this addition of the P and the N nucleotides, you're building up this random sequence at the junction of these VDJ segments. So that's giving you that extra variability. And this is just summarizing that in a little bit more detail. Well, not more detail, it's just kind of highlighting where we get the addition of the P nucleotides, so the palindromic, and then the N nucleotides to generate that kind of random junction. And then, of course, we have this combinatorial diversity of the antigen binding site. So this really just means that once we've generated our heavy chain and our light chain, we can bring, a heavy chain together with a particular light chain that will give that kind of extra variability of the antigen binding site. So we've got the diversity in bringing the different VDJ segments together. We've got the junctional diversity in the little junctions between the VDJ segments that gives us that extra variability. And then we've got the option of putting a heavy chain with a particular light chain, and that gives us an added layer of variability. And then we have somatic hypermutation that we'll talk about later, so don't worry about that just now, but that can give us further sort of variability that's quite important. But that only occurs once the B cells have been activated. So this is kind of what we're looking at today is what's going on in the bone marrow. So that's all well and good, but we really want to ensure that once we've successfully produced a unique antigen receptor or immunoglobulin that's on the surface of the B cell, we need to stop that process because we want to make sure that that B cell only has one sort of antibody. It's only got one membrane-bound receptor. And this is mediated by allelic exclusion. So once all the gene segments have been put together, we essentially need to shut down this process. So you can see here, this diagram is just defining allelic exclusion, which essentially means that once we've got that successful immunoglobulin on the cell surface, everything is shut down. But you see here that we've got our B cell, we've got our recombination events going on for the heavy chain. If we don't get successful recombination, the cell can try again, and if we don't get it a second time, it can try again, after which it's eliminated by apoptosis. If we get successful VDJ recombination, we can then get our light chain recombination events occurring. And likewise, if it doesn't work, the cell can try again. If it still doesn't work, it can switch from the kappa light chain locus to the lambda light chain locus, give it another go. If it doesn't work, it can have another go. If it doesn't work, then it's eliminated. But once it has produced a successful immunoglobulin, that's it. The process is shut down so that we've only got that receptor of that one specificity expressed on the cell. Okay? If we look at the T cell receptor, so this is obviously the structure of the T cell receptor. So we're just going to have initially a look at the structure and then a look at how we can generate that diversity of the T cell receptor and have a look at the functions and some of the key differences. So this is just comparing the T cell receptor to the B cell receptor. So you can see that they're somewhat similar, but obviously the T-cell receptor is a little bit more simplified. The T-cell receptor, instead of having a heavy chain and a light chain, it has an alpha chain and a beta chain. It's generated by the same gene rearrangement mechanisms that we saw for the B-cell receptor. This enables it to have, be very variable and have a high diversity, and this gives it a sort of unique antigen specificity. And again, it's monospecific for each T cell, so we only have one sort of T cell receptor on the T cell that's given for a specific antigen. Remember again that T cells are recognizing peptides that are derived from pathogen proteins, so it's not the whole protein. So for a B cell receptor, that's recognizing the whole protein, but for the T cell, it's just recognizing peptide fragments that have been presented to it. So when it comes to the structure of the T cell receptor, you can see it here with the alpha and the beta chain. It's also co-associated with a CD3 co-receptor, so that's an important kind of receptor to remember, the CD3. Again, this mediates that intracellular signaling events from antigen binding to the T cell receptor, helps mediate it across the plasma membrane. So, as we mentioned before, the T cell receptor, it just has one antigen binding site that you can see here. Its structure kind of looks a little bit more simpler, but essentially the alpha chain is similar to the immunoglobulin light chain, and it just has VJ segments, whereas the beta chain is similar to the heavy chain from immunoglobulin, and this has VDJ segments. Again, it's a similar process, so we've got this combinatorial diversity, junctional diversity, and then we're getting the combinatorial diversity by adding together the alpha and the beta chains. But once this is generated to form a T cell receptor, that's it, it's fixed. So unlike the B cell receptor, which has that potential to undergo somatic hypermutation and further change once we get binding of antigen, that's it for the T cell receptor. And this just shows the process of VDJ recombination for the T cell receptor. So you can see for the alpha chain, we've got the V and the J segments coming together in a constant region to form the alpha chain. And then for the beta chain, we've got the VDJ recombination coming together to form this VDJ segment with another constant region. And this is all occurring within the thymus. Now, when we talk about T cells, we will talk about T cells being alpha-beta, okay? And then they're called alpha-beta T cells because of the alpha and the beta chain. But I just want to make you aware that there is another class of T cells that are called gamma-delta T cells. And this is because they have a gamma chain and a delta chain. So we're not going to go into much detail, but I just want you to be aware of that. The gamma-delta T cells, they're a smaller population, they have distinct functions, and a lot of research is still being done on them. But when it comes to the gamma-delta chain, their locuses, they have basically different locuses. So the delta locus is located within the alpha locus on chromosome 14, and the gamma locus is located on chromosome 7, and it kind of closely resembles that of the beta locus. But just to make you aware that there are different T cell receptors out there. It's not just alpha-beta, but generally when we talk about T cell receptors, we'll be talking about the alpha-beta ones in these lectures. So in terms of just pulling things together of what we've talked about so far, so essentially the whole story of antibodies and T cell receptors and this whole gene rearrangement was really one of the main advances in immunology that we've seen so far. And it's really shed sort of an important light in how we mount an immune response to a pathogen that we've never seen before. So if you think about a pandemic, something that we've never seen before, we have that ability to recognize a pathogen that the human race has never encountered. And that's because of this whole process that we've been talking about today. Now, once we discovered this process, the actual mechanisms by which these gene segments are put together is just as important. And that's a whole area of research as well. But by understanding how we get this arrangement and this rearrangement of these genes, we understand a lot more about how our T cells and our B cells work, both normally and abnormally. And we've got a little time for a quiz, I think, if you just want to have a go at that. So there's just a few questions, a little bit more picture-based today. So it should be quite quick. So click on the T cell receptor. This one is the T cell receptor. This is the B cell receptor over here. So T cell receptor looks like the majority of you got it. Click on the B cell receptor. It's this one. A little bit of a trick. This is antibody. So we're looking at the B cell receptor because it's associated, it's membrane bound. Okay. Click on the heavy chain. The heavy chain is this one here, or this one, and these are the two light chains. In terms of the antigen binding site, that looks great. The FC region is this portion down here. So these are the Fab fragments. This is the FC region. B cells recognize antigen in its native form. That's true. T cells, recognize peptides of proteins. That's true. And when it comes to clonal expansion, it's the expansion of a single cell clone following activation and the proliferation of activated lymphocytes. So those examples look good as well. Okay, so that's it for today. And then on the lecture on Thursday, we'll start to have a look at the major histocompatibility complex with a view to antigen presentation. So I'll see you guys on Thursday.